Hello, welcome to today's video. It is on internal membranes and enzymes. The learning goals for today are to explain, using an example, how the arrangement of internal membranes can control biochemical processes. To recognize that biochemical processes are controlled and regulated by a series of specific enzymes. To describe the structure and role the active site on an enzyme, and to explain how reaction rates of enzymes are just affected by different factors, and we will look at temperature, pH, inhibitors, and how concentrations of reactants and products play a part. So you can pause the video here and just write a summarized version of those goals. Cool, let's get into it then. So from 3.1 of your textbook, we have to explain how the arrangement of internal membranes has an effect. And I want to start by saying, why are eukaryotes different from prokaryotes? Well, there's a number of differences between them, as you know. Firstly, they're more complex and they're larger, and one of the main reasons why they are able to be more complex is because they have membrane-bound organelles. So organelles such as the mitochondria that have their own little environment, or lysosomes, these little packages of proteins that have their own thing going on inside of them. Prokaryotes don't have that feature. So compartmentalizing a cell by having membrane-bound organelles creates specialized environments for specific functions. This enables a large number of activities to occur at the same time in a limited space and under different conditions. For example, lysosomes, they're also known as the garbage collectors of cells. They contain strong acidic enzymes which provide an environment to break down foreign or damaged material within a cell. Other organelles, such as the mitochondria, they increase their internal surface area by folding and stacking this membrane inside of them. They contain two membranes for this to occur. They first have what is called the smooth outer membrane, but then they also have a folded inner membrane, like a folded, crumpled up bag within an outer, smoother bag. The inner folded membrane forms what we call cristae. These are folds that simply increase the surface area, and more surface area means more reactions can occur within the mitochondria. The cristae is studded with enzymes and ribosomes that provide the sites for chemical reactions to take place. The mitochondria is the site for cellular respiration, and that is when we take in oxygen and sugar and ultimately produce ATP, the energy for the cell and for the body. The space within the folds, in between them, is called the matrix, and it's this gel-like substance enclosed by the inner membrane of the mitochondrion. The folding of the cristae means no part of the matrix is far from the inner membrane, so therefore it's just a little distance between every part of the cristae and the matrix, and that just facilitates and enables diffusion to occur. Here is an image from a scanning electron micrograph of a mitochondrion in a nerve cell. So you can see in this real life mitochondrion that there's all these folds called cristae studded with ribosomes where cellular respiration is taking place, and then there's the matrix in between them. That is the inner membrane enclosed by the outer, smoother membrane. That is an example of how the arrangement of internal membranes can control biochemical processes. Cool, so let's keep moving on. Enzymes and pathways from 3.2 of the text. So you may have heard of the term catalyst before. A catalyst is a substance that speeds up the rate of a chemical reaction but is not itself changed by the reaction. So enzymes act as biological catalysts, as in they occur within our body, within our cells. Enzymes are proteins. We have two examples here, catalase and amylase. And they are molecules that are folded into a specific shape for a specific function. To help you remember enzymes, they often end in ase. Enzymes are chemicals that aren't consumed in a reaction, as we said, but they simply speed it up. One example that we will talk about is catalase, which is found in basically all living cells. What it does is it breaks down hydrogen peroxide, that's H2O2. It's also commonly known as bleach, and in dilute concentrations is used in the household for cleaning and also in dyeing hair. But in its proper form, like in a concentrated form, it actually kills cells. Like if you touch it, it'll burn your skin. It will also be produced naturally within the cell. So in chemical reactions, it builds up. So our cells need to get rid of that before it becomes too toxic for our body. And so we use the enzyme catalase to help in that. Catalase breaks hydrogen peroxide down into H2O and oxygen. It does this at an incredible rate. They believe that there's about 40 million molecules of hydrogen peroxide that go into a single catalase enzyme and are broken down into water and oxygen every second. So let's zoom out again and talk about what do enzymes do. Basically, they control what we call biochemical pathways. So they are chemical reactions in cells that occur in a series of steps, which are regulated by enzymes. Enzymes are important because without them, reactions just would take place, but at such a slow rate, and that rate would be incompatible with life. So this diagram here from the textbook, it shows a biochemical pathway, and the products or outputs of the first step, they become the reactants for, or the inputs for the next step until the final products are reached. So each step is regulated by a specific enzyme, and what this does is it just saves products or things just accumulating within the cell, and when things accumulate, it just becomes too busy and we can't get done what we really need. So it's kind of clever how the body automatically it just does this. It uses products to become reactants. It's just being efficient. So enzymes are used in vital metabolic reactions, and basically cellular metabolism simply refers to all of the chemical reactions that take place in a living cell to facilitate life. So enzymes, our learning goal is to recognize that biochemical processes are controlled and regulated by a series of specific enzymes. And one example is how hydrogen peroxide is a output of metabolism, but it is broken down by catalase. Now this process would actually take place by itself. If I had a container of hydrogen peroxide and I left it on a bench, it would break down spontaneously into water and oxygen, but it would take many, many years, thousands, if not millions. Yet in the presence of catalase, it takes place in moments. So enzymes are absolutely essential to sustaining life. Let's move on. So how do they actually work? We're going to zoom in a little bit now to the anatomy of an enzyme. We're going to define the structure and the role of an active site. We will use the example, like how does catalase actually break down hydrogen peroxide into water and oxygen? Basically, it has what is called an active site. And it's this part of an enzyme that has this hole in it, which the substrate is going to fit into. So going back to catalase, it is the enzyme. Catalase is the enzyme, and the substrate is what goes in. So that would be hydrogen peroxide. Enzymes basically tug on the substrate to break them down. Each enzyme is specific to one substrate or molecule or type of molecule. So you can see, like, from this particular enzyme, there is a specific shape, something that's going to fit into it. And the term for this is specificity. There are two prominent models describing how substrates can actually bind to enzymes. There is the lock and key hypothesis, and there's the more recent induced fit model. So we're going to talk about those. The lock and key hypothesis essentially states that the active site specifically matches the shape of the substrate molecule, like a key going into a lock. It has this specific shape, and if it doesn't have that shape, then it's not going to fit, and the enzyme isn't going to do what it needs to do. Still, this shows that the substrate binds to the enzyme, then tugs on it, creates this enzyme product complex, and then those products are produced. The other model is the induced fit model. And this says that enzymes' active sites actually undergo changes in shape to better fit the substrate. And you can see here that it's not the exact same shape, but it will still go in there, and then the enzyme will kind of mold to fit it to produce the same outcome, the products. So this was a more recent understanding of how enzymes work. This was an earlier understanding and framework, and the induced fit model is more widely accepted and taught now. So again, considering hydrogen being broken down, hydrogen peroxide, into oxygen and water, as we said, that will take place naturally, but it just takes so long. It is in the presence of catalase that it happens quickly. And this happens because the enzyme decreases what we call activation energy. That is the energy required to initiate the reaction. So if you look at this diagram here, there's a bit going on, but what it shows us is two lines. On our axes, we have energy, and we also have reaction progress. So let's look at, firstly, the darker shade line. And here we can see that there is energy needed for the reaction to progress. And it's actually quite a lot. But once it gets over that energy that is initially needed, then the reaction can progress and the products are formed. So that amount of energy to get the ball rolling, so to speak, is called the activation energy. And without an enzyme, it's actually very substantial. But the same reaction in the presence of an enzyme, not so much energy is needed because the enzyme has decreased that activation energy. So the reaction can take place more readily and easily in the presence of an enzyme. And that is how enzymes basically work. They decrease, again, what we call activation energy. So if we move on now to 3.4 and talk about factors affecting reaction rates. As we said at the start, there's a number of factors, including temperature, pH, inhibitors, and just the concentration of things. So to begin with, let's talk about temperature. At low temperatures, enzyme-controlled reactions go slowly because the molecules have low kinetic energy. But when temperature increases, the reaction also increases as molecules are moving around. And basically, they think that substrates are just flying around within a cell, and when they find an enzyme, that's when they bind. And so when you have more temperature, more heat, then those substrates are moving around at a greater rate, so there's a greater chance of a collision basically taking place, that substrate finding that enzyme. Now, that works up until a particular point. There's usually an optimum temperature, and that's about 40 degrees Celsius. That's the temperature that our bodies run at. But in our body, if the temperature increases above that optimum temperature, then those enzymes can actually become what we call denatured. And denatured just means that enzyme has been affected by heat too much, and it's actually changed shape and no longer will do its job. This is why like fevers and temperatures can actually be quite dangerous. It can lead to death. The enzyme, once it's changed shape, can't ever go back to what it was. So in this diagram here, if we just look at temperature for a bit, we have enzymes within a human intestine functioning at about 37 degrees. That makes sense. That's our core body temperature. But then we have different type of enzymes not within our human bodies that function at their own optimum temperature. For example, chlorophylls found in the Arctic, where it's very cold, their optimum temperature is a lot lower. Whereas enzymes found in the geothermal seabed, they have a much higher optimum temperature. So that is how temperature affects enzyme activity. We also said that pH does as well. So again, enzymes prefer to work at an optimum pH. Outside of its pH range, whether it's above or below, the enzyme is again denatured. And here from the textbook, we can see the pH scale, rate of reaction up this side, and acidic and basic on the two opposite ends. We can see that there are 3 different enzymes here, and they basically have their own optimum pH range. So human pepsin in the stomach, where we know it's extremely acidic, there's hydrochloric acid being produced, operates in the very acidic range. That makes sense. Whereas the blue line represents in our bone, where it's actually quite an alkaline environment, and the human salivary enzyme amylase, which breaks down what we chew and what we eat, it has its optimum pH range of about 7. So it depends on the environment what the optimum range is. And lastly is concentration. So it says that the concentration of reactants and products can also affect enzyme activity. And reactants are what are going into the reaction, and products are what are coming out. So we're talking in terms of a series of steps, a biochemical pathway in this part now. So substrates and enzyme concentration can also affect reaction rate, because the more substrate there is, the more reactions that can take place until all the enzymes are working at working capacity. So that makes sense. The more substrate there is, the more that can take place until like all the enzymes are being used up, basically. Then it reaches what we call saturation point. The more enzymes there are, also the more product that is made, until the products begin to become too many, and then they can just over accumulate and basically start to inhibit enzyme activity. And if you remember back to if substrates are flying around trying to find an enzyme to collide with to do the reaction, well, if there's a whole bunch of products floating around as well, then it just decreases their chance of finding that enzyme. So there's this kind of balance that needs to be found. And the body actually has magnificent ways of finding that sweet spot. They can turn on enzymes and turn off enzymes to maintain proper concentrations of substrates and products. And so we're going to talk about in a moment inhibitors, how enzymes can be turned off. But in your textbook, you will read about cofactors and coenzymes. And they're basically these molecules that just assist in turning an enzyme off. And sometimes without them, they can't do what they need to do. So it's our way of, again, regulating what needs to take place. Regulating when we don't want a reaction to take place comes in inhibitors. So there are two types of inhibitors, and basically they will stop an enzyme from doing its job. We can have a negative scenario and a positive scenario. Sometimes poisons can actually come in and inhibit our enzymes from doing our job, and that can obviously cause death. But then we also have examples of when we want to turn off our enzymes. So there's two types. We have a competitive inhibitor and a non-competitive inhibitor. And a competitive inhibitor basically has the same shape as the substrate, and it wants to join onto the active site, inhibiting the substrate from joining that active site. So they both compete over the same site. Whereas a non-competitive inhibitor, it does not bind to the active site. It binds somewhere else on the enzyme, but in doing so, it stops the reaction from taking place, even though the substrate still binds. If you'd like to know more about where this takes place in the real world, are research applications and uses of inhibitors. Check out competitive and non-competitive inhibitors and how they're used in the medical world, in the drug world. And also some other examples where, poison can come into our bodies and we don't want that, but it's still very interesting. So, moving on, here I would like you to pause the video and just practice, talk it over in your head. How do enzymes work? Go. Sweet. So hopefully you just pause the video and talk through a process, something like this. So say you've got an enzyme and it has an active site. Then a substrate comes along and it binds to the enzyme. Once it is bound, it creates this enzyme substrate complex. Depending on the model that we're talking about, if it's the lock and key, it's a perfect fit. If it's the induced fit, then the enzyme will kind of mold itself to fit the substrate. Reaction takes place and out come the products. An example is being hydrogen peroxide coming into catalase, which is then broken down into oxygen and H2O. So, homework for this week. To read chapter 3, to add key terms from this video and from your readings to your glossary, and I'll also be handing out practice data test revision sheets. So those are super useful and important for you to complete. If you'd like support, check out the Amoeba Sisters video on enzymes. Even though we've watched in class, depending on the class you're in, it's still very good to watch over. See how you go with the readings. If you need to go light on them, just looking at key terms and looking at the images, so that you can focus on data test revision sheets, then do so. Let me know how you're going if you need to. So to finish up, just a couple of gifts from the Amoeba Sisters. Here we have an enzyme and a substrate, and they fit in what is called the active site. And one more. This is the induced fit model, and I think they call it's just like an enzyme substrate hug where it just molds. And that is all for this week. I hope that you have found this lesson interesting and helpful, and I look forward to seeing you guys in class. God bless.